Hi, everybody. I'm Julie Harris. I'm the Associate Director of Neuroanatomy at the Allen Institute for Brain Science. It's my pleasure to introduce to you our first Institute team talk of the showcase event. This is the Synaptic Physiology team. Their talk is going to be revealing dynamics in the cortical microcircuit. The success of our large-scale projects are due really to successful interactions of many individuals, as you've heard now from Jim and Saskia and also from Alan. And these are individuals with various different roles in the lab, from kind of data generators in the lab, I should say, who are actually hands-on collecting the data, to people that are responsible for data analyses on the other end of the pipeline. So representing these different roles for the team today are Luke Campagnola, Alex Hogarth, Alice Basma-Moody, and Jung Hoon Lee. So welcome. Thank you.
Thanks, Julie. We've lost notes up here, or anything. Thank you. Okay, thank you all for joining us this morning. I just want to start out by talking about some of the Institute's broad goals and where synaptic physiology fits into that picture. So we want to understand how the brain works. And I think that as a field, we are incredibly fortunate to be doing neuroscience today. Of all the truly fundamental problems in science, I think that only a few are really left unsolved today. Intelligence is one of those problems, along with the grand unified theory of physics and the origins of life. It's a really historic time to be doing this work. And also why projectors are still difficult. Okay, so this problem is way too big for us. So as you can see, we narrowed it down a bit. We're just going to focus on the cortex here. Okay, but we're going to break it down even further into cortical subregions and microcircuits, and we go all the way down to the level of individual cortical neurons. This seems like a pretty good place for us to start, so we're going to build back up from here. We have many different types of neurons. Each type somehow performs a unique function in the circuit, and they're all connected together in a predictable way. And we hope that by carefully cataloging all of these individual pieces, mapping out how each cell type behaves and how they talk to each other, that then we will stand a better chance of understanding how the system works as a whole. I think almost everybody in the Allen Institute for Brain Science is contributing to some part of this picture. In synaptic physiology, we're focused on this bit of the picture. This is a synapse. They are tiny but surprisingly complex machines. It's a pretty broad audience today, so we're going to do a quick crash course on synaptic physiology. First, a synapse is the point of communication between two neurons. We call these the presynaptic and the postsynaptic. Electrical spikes, these action potentials in the presynaptic cell, can evoke these tiny little electrical blips in the postsynaptic cell. This is the entire basis for communication between neurons. We call these postsynaptic potentials, or PSPs. In almost every case, the identity, the type of the presynaptic neuron determines whether these PSPs are going to be inhibitory or excitatory. And finally, synapses are dynamic. And what I mean by that is that the strength of these PSPs, their amplitude, can change from moment to moment. So I'll give you two simple examples. If we look on a very short time scale, a train of spikes in the presynaptic cell can give you a situation like this, where you've got PSPs that are slowly increasing in amplitude over time. And we would call this short-term facilitation. In contrast, we've also got synapses that do the opposite. They get slowly weaker over time. We call this short-term depression. And this is particularly important because it gives the synapse the ability to process information. It doesn't just relay signals from one cell to the other, but it actually does a lot of filtering. So we have many different cell types in this microcircuit, and they're connected by synapses with a diversity of different functional properties. There's actually a huge number of these cell types, but in order to simplify the problem again for the purpose of our experiments, we're actually just focusing on these broad cell classes. So when we do these experiments with human tissue, that means that we're classifying these cells based on their morphology, on their location in the cortex, and on their electrophysiological properties. With mice, we also have the option of transgenically tagging specific cell lines with fluorescent markers. So for example, I'm showing a few of our favorite cell classes here. SIM1 and TLX3 are transgenic lines that mark layer 5 pyramidal neurons, whereas somatostatin and parvalbumin are two broad classes of inhibitory interneurons. So our goal in synaptic physiology is really to map out this relationship between cell types and synaptic properties. Two simple examples of this idea. Synapses from these parvalbumin neurons onto TLX3 are inhibitory. They're relatively strong. This is about 0.5 millivolt here, and they depress over time. They get smaller. In contrast, if we look at a synapse from TLX3 onto a somatostatin interneuron, they're excitatory. They start out a bit weaker, but then they facilitate over time. Okay, so this idea of mapping from cell classes onto synaptic properties is not new, but it's really taken off in the last several years. There's been this explosion of articles generated that each describe some portion of the circuit. And this is fantastic for us, but they all describe different parts of the circuit in different ways, with different methods and in different experimental conditions. So that makes it very difficult for us to construct a big picture view from all of these disparate pieces. So we're at a moment in neuroscience where we have an opportunity to make a big impact here. And we want to do this the Allen Institute way. We're doing big team pipeline science to make sure that we characterize everything as completely and consistently as we can. Okay, so I'm going to switch gears. I'm going to talk about the techniques that are available to us for just a couple of minutes. There's no single technique that can really extract all of the properties that we want to know about synapses. So we have multiple groups at the Institute working on this problem in parallel. We've got electron microscopy, who you'll hear from later on today. We have array tomography. We have various tracing projects, transsynaptic tracing, many different techniques. Most of these use some kind of imaging, which gives us a snapshot in time of what the synapses and the circuits look like. But as I mentioned earlier, these synapses are dynamic machines. So we also need a way to probe the synapses in a living system. And that's what synaptic physiology is for. We're using a very old and simple technique called patch clamp electrophysiology. The idea is pretty simple. We start with a piece of living brain tissue. We look for a couple of neurons. At this point, we have no idea if they're connected. So we attach a couple of microelectrodes to them. And then we can use one electrode to force this neuron to fire action potentials. And then we basically just record from the other neuron and find out if we see a response. If we do, then it's connected. Great. And we also have the option of playing with the timing of these spikes so that we can actually really probe the dynamics, the dynamic behavior of the synapse. The downside to this approach is that it's really slow. And it's pretty difficult. These electrodes are difficult to work with, and each synapse that we're looking at one at a time takes about 45 minutes to characterize. So we have a few different ways that we can speed it up. One is really simple. You just add more electrodes. And if you get up to 8 electrodes, then in an experiment like this, you can probe up to 56 connections in parallel. So that speeds things up reasonably well. And we built a pipeline out of this technology. We've got a few of these Octopatch rigs that have been collecting data for almost a year at this point. In a few minutes, Alex is going to tell you all about the work we've done there. But first, I want to tell you about one more technique we have to speed this up. I mentioned that these electrodes are really difficult to work with, and that's a big part of what slows us down. So very recently, new transgenic tools have allowed us to stimulate these neurons to fire action potentials using lasers instead of electrodes. And what this allows us to do is to replace that presynaptic electrode with a laser that can rapidly scan and activate many hundreds of neurons in the course of a single experiment. Did we miss something? OK. So coming up very soon, Alice is going to tell us about the optogenetic experiments that I just described, followed by Alex, who's going to talk about multipatch. And at the beginning, I mentioned that what we really want to be able to do is to take all of these experiments and build up a picture of the way the system works as a whole. And so Jung is going to take us in that direction and talk about what happens with our data after it leaves the pipeline.
All right, thank you, Luke. So I'd like to start with just an example of what these optogenetic experiments look like. So here we're looking at a slice of cortical tissue. These red cells express light-sensitive proteins called opsins, and I have 2 electrodes here recording from these green cells. In this very specific example, we can shine our laser on this purple cell right here. It causes it to spike, and because it's connected to this cell, we see a small synaptic response. Similarly, we can shine our laser on this cell, and again, we'll see another small synaptic response. Note that both of these connections are excitatory. Whereas this cell right here actually provides inhibition to both of the cells I'm recording from, although with different strengths. So these are exactly the types of connectivity patterns we're hoping to uncover using this technique. But in order to achieve this type of experiment, there are a few important technical hurdles to overcome first. If we are to actually locate connected neurons on the single cell level, our light stimulus needs to be able to work on just one cell at a time. But brain tissue is a really complex 3D web with neurons all clumped together and sitting on top of one another. So for example, if we want to test to see whether this very specific green cell is connected to this blue cell, our light stimulus needs to cause this green cell to spike. But it can't also cause that green cell to spike, or that green cell to spike, or the cell below it to spike. Otherwise, we just confuddle our results. So this is where 2-photon laser stimulation comes into play. 2-photon lasers allow us to precisely target light in tissue. We can take a small, about 1 micron beam and scan it across a single cell body. In this way, the stimulation is restricted to the cell we care about in X, Y, and Z. So now we have 2 requirements. Our cells must be sensitive enough that this different type of laser scanning photostimulation will actually cause them to spike, but they cannot be so sensitive that they spike when we target a nearby cell. We can call these our sensitivity and our specificity requirements. So we started by working in close collaboration with the mouse genetics team here, the transgenic team here at the Institute, to test the responses of different transgenically expressed light-sensitive opsins to simple wide-field LED stimulation. We tested 4 opsins: Chronos, channel rhodopsin, Reacher, and Crimson. And we can see that these last 2, Reacher and Crimson, have significantly larger and slower photocurrents when compared to Chronos or channel rhodopsin. So this hints to us that both Reacher and Crimson may be good candidates for our laser scanning stimulation, but we need to be sure that we can actually reliably evoke spikes in these cells with 2-photon scanning stimulation. And indeed we can. So as we increase the power of our stimulus here, you can see the cell begins to spike. And while that's promising, it is just one cell. So we repeated this test across many cells and found that we could actually achieve spiking in all cells given the proper choice of power down here. Additionally, we also expressed Reacher via a virus that localizes to just the cell body of the neuron. That's this green line right here. And we could also achieve 100% spiking. So this just puts more tools in our toolbox to be able to perform these connectivity mapping experiments. So now we have 2-photon sensitive opsins that elicit spiking. But that brings us back to our specificity requirement, that our opsins cannot be so sensitive that they spike when we're targeting the neuron next to it. So we did a series of experiments to measure the resolution of our laser stimulation. In this example, we're stimulating 2 adjacent cells, and we found that stimulating this cell on the left here has no effect on this cell on the right. And we've also done similar experiments where we varied the location of that target stimulation in X, Y, and Z. This lets us know that during a real mapping experiment, when we target a very specific neuron and we see a connection, that connection we found is coming from the neuron we targeted and not the neuron next to it or the neuron below it. So by now, I've hopefully convinced you that our method of photostimulation is both sensitive and specific. And with this groundwork in place, I can show you the results from some of our early connectivity mapping experiments. So we started by measuring excitatory to excitatory connections in layer 5 of the primary visual cortex. This image is an example experiment where we're recording from this green cell here, and we're stimulating all of these red cells expressing Reacher. And over here is the result of repeated stimulation to this single target right here. And the average in green is a pretty convincing synaptic response. And the map down here is actually an aggregate across slices and across subjects, all aligned to the excitatory neuron we were recording from in the center. So this is exactly the type of data we're hoping to collect using this new technique, the probability of connection between cell types and the spatial distribution with which these cells are connected. And as you can imagine, we're not only interested in excitatory neurons in layer 5, and we can also look at connections from inhibitory neurons to excitatory neurons. So again, in this example, we're expressing our opsin reacher in green GAD positive interneurons and recording from this excitatory cell in red here. Again, you can see example connections over here, which is a result of repeated stimulation to the targets 1, 2, and 3, and another aggregate map down at the bottom showing the spatial distribution of connections onto excitatory cells. And in the future, we plan to conduct similar experiments targeting opsin expression to a variety of cell classes and looking at connections within and across layers. So to conclude, I have shown you that we have successfully developed and utilized a novel photostimulation technique to conduct circuit mapping experiments and contribute to the goal of understanding the cortical microcircuit. But because we have the potential to test so many connections, we are unable to fully characterize each one. So now Alex will tell you about a complementary multi-patch technique we are using that provides a characterization of both synaptic connectivity and dynamics.
All right, thanks, Alice. So I'm going to be talking about some of the work we've been doing on the multi-patch electrophysiology pipeline, looking at synaptic connectivity in both human and mouse brains. So I'd like to start by walking you through an example of what a multi-patch experiment might look like. And so here we're using transgenic mice with 2 classes of fluorescently labeled cells. You can see the TLX3 pyramidal cells are labeled in green, which you can see from their characteristic morphologies on the left. And the somatostatin inhibitory interneurons are labeled in red, which you can see below. So each of our pipettes contain a blue dye so we can visualize our cells and confirm their identity after the experiment. And so in this experiment, we're going to be recording from both TLX3 pyramidal cells as well as the somatostatin cells. And so in each individual experiment, we elicit a spike in one of our cells by delivering a current pulse in one electrode. And we record the electrical activity in each of the remaining cells immediately following that spike in order to look for a potential postsynaptic response, which would show up in the voltage trace in each of these cells, which you can see on the right. And so next we alternate delivering spikes to each of our recorded cells and look for postsynaptic responses. And so here you can see that a spike in cell 4 has driven a response in cell 3. And so we can draw in that synapse. And so next, when we also include the somatostatin cells, we can look for connections across these cell classes. And so here you can see 2 more excitatory connections onto somatostatin cells and 1 inhibitory connection from a somatostatin cell back to a pyramidal cell. And so we can draw in our cortical microcircuit. And so because we're both stimulating and recording from the pre- and postsynaptic cell, we can look for reciprocal connections like the one between cell 2 and cell 8. And because we have precise control over the presynaptic spike time, we can deliver trains of spikes at different frequencies and look at how these responses evolve over repeated stimulation. And so in this same inhibitory example from the SST to the TLX cell, you can see that this response is depressing. If you compare the response to the first spike to the response to the 8th spike, it gets smaller. And so next I want to highlight that we're also able to do these experiments in surgically resected human cortical tissue. And so here we're looking for pyramidal cells and filling with a green dye. And you can see that we were able to find multiple excitatory connections between these pyramidal cells in layer 3 of the human cortex. And so what's really interesting about these experiments is eventually we're able to build up a set of rules which are governing the cortical microcircuitry in the human cortex and compare the connectivity and the synaptic dynamics across these species. And so these synaptic connections can be fairly small and difficult to detect, like this example you can see here. And so it's important to consider whether or not we're able to detect a connection. So for each given response, we plot the signal strength against the background noise. And in order to confidently state that a connection has been found, we want to see a high signal and relatively low noise. If there was a recording which falls below this diagonal line, we might as well post those on Craigslist because they're going to be missed connections. However, the vast majority of our recordings are up here in the detectable range. And so with this criteria, we can take each experiment and bin our connections by pre- and postsynaptic cell type. And you can see even within this single experiment, different synapse types may be more likely than others. And so once we've done this for a sufficiently large amount of data from a number of experiments, we can categorize each synapse type by a probability, which is given by the total number of connections we found and the total number of synapses that we've probed. And this results in our synaptic connectivity matrix, where the presynaptic Cree type is on the left and the postsynaptic Cree type is across the top. So these boxes in gray here are experiments we have yet to carry out, and the boxes in blue are synapses we have yet to find. But so if we do this for each of the Cree types that we've studied, we can come up with a probability of finding a connection. So for example, from this parvalbumin cell onto the SIM1 cell, there's an overall probability of about 36%. However, we're not sampling from every single connection in the population, And so this only gives us an estimate of true connectivity. And so each matrix element also has an associated confidence interval, which gives us a sense of the certainty of finding a connection. And so these data represent over 500 detected connections from 6,000 probe potential synapses and rising, with an 8.2% overall connectivity. And so each matrix element actually contains more information than the raw likelihood of finding a connection. The individual synapse types themselves have unique characteristics. So even if we look across a single given row of the matrix, we can find individual cell types have heterogeneous properties depending on their postsynaptic partners. And so again, looking at the parvalbumin cell, we see a diversity in the strength and the kinetics of the postsynaptic responses looking across the 5 major cell classes in layer 5. And so using this technique, we were able to build out a catalog of responses and find and characterize responses between the 3 major inhibitory cell types in the cortex, as well as between the 2 major excitatory pyramidal cell types in layer 5 of V1. And so for the first time, we can begin to systematically characterize these synapses in a Cree-type dependent way. And so we've also started characterizing differences in synaptic strength and short-term plasticity. As you might expect, there's a huge diversity in the dynamics, and they varied across these synapse types. And we found facilitating and depressing synapses and everything in between. So it would be really nice to be able to represent each of these synapse types with a single concise model that captures their diversity and covers the range of the responses we've been observing. And so that's what Jung is going to tell us about now.
Thank you, Alex. I will discuss the synapsis model we are currently developing. So our data confirm that synaptic transmission is non-stationary. So it can facilitate or depress. Then, why does the brain need such short-term synaptic plasticity? Earlier theoretical study proposed some possible answers. For example, York and Rodham showed that depressing synapses can induce the sequential activation of neurons or propagating bump. And this propagating bump can account for place cell activity observed when animals are resting. Also, facilitating synapses can serve as working memory. So we asked, what are functions of short-term synaptic plasticity in visual perception? To address this question, we are building synapse models that can capture short-term synaptic plasticity in V1, and we will incorporate this synapse model to the network model of V1, which are being developed simultaneously. The starting point of our synapse model is T-sorting macro model. In this TM model, the depletion of vesicles and the increasing release probability can account for depression and facilitation respectively. And these 2 simple equations can describe them pretty well. However, the actual synaptic transmission is much more complicated than this TM model. The speed of replenishment can be history dependent, and postsynaptic receptors can lose their sensitivity. Also, release probability can change very slowly. This use-dependent replenishment, desensitization, slow modulation of release probability can be described by these 3 simple equations. So our model considers these 5 gating processes. In the experiment, to measure PSP, we stimulate presynaptic neurons using spike trains at fixed frequencies. Not surprisingly, we found that synaptic transmission is very noisy. As you can see here, PSP traces can be highly variable in some cases. If synapses are homogeneous, we can average them to reduce the effects of noise. However, they are not homogeneous, even when they share the same pre- and postsynaptic neurons. This figure shows the PSP ratio, and this horizontal line is the boundary separating facilitating and depressing synapses. Synapses above this line are facilitating. Synapses below this line are depressing. So for each synapse type, we used clustering algorithms to identify homogeneous synapses first, then calculate the average PSP. This is an example of our data between PV neurons. So we first measure the PSP evoked by the first pulse of a 200 Hz spike train, and we use this value as a reference point to normalize all other PSP. This is the PSP evoked by second pulse obviously this synapse is depressing. We played 8 pulses first, then inserted the delay. After this delay, we played 4 more pulses to examine the recovery as well. After that, we fitted these 12 points to the model. In the actual experiment, we used multiple stimulation frequencies, not just 200 Hz. In this figure, different colors represent different stimulation frequencies. They look messy, but a single synapse model can describe them pretty well. We found that the depression and use-dependent replenishment are necessary to account for this type of connections. The parameters we found are shown in this table. So far, we have developed a network model of V1 to study functions of cell types only. And this figure shows one example of our models. But in the future, we will incorporate these synapse models to this type of network model of V1 to study functions of short-term synaptic plasticity as well. So, look.
Thanks, Jung. As you might imagine, we have a lot more work to do to generate enough data to really fill the models that Jung is describing. So we're continuing to expand our catalog of synapse types. And we have several other groups, as I mentioned, at the Institute who are working on this problem in parallel to produce complementary data sets. You'll get to hear from one of these groups, I think, after the break. It takes a huge amount of support from many, many people to make experiments like this possible on a large scale. So I want to thank all the many groups that have participated here, and especially the folks in synaptic physiology who you didn't get to hear from today.
So we have time for some questions, and if you raise your hand, somebody will come to you with a microphone.
So this is a great, the question is looking at PV and somatostatin interneurons as cell types and whether we're going to subdivide those. And the answer is absolutely yes. PV and SST for us are just broad cell classes. We use these in these experiments to simplify the problem for ourselves. But there are other groups at the Institute that are breaking these down into much finer cell classes, cell types, transcriptomically in particular. And we're hoping to incorporate some of that work into our own experiments using multiplex FISH later on, but that's still in development.
Yeah, on your array of connectivity, you had some cells that were 0 out of 36. Some were 125 out of 501, 35 out of 1,000. So it's a really wide range of apparent connectivity that you're observing. So 2 questions. One, what's the actual number of connections you expect to find in any of those cells that you're reporting on, if you were able to find all the connections? And number 2, with this diversity that you're finding and trying to build into the model, are you beginning to learn any architectural principles from this small subset that you've been able to study?
Okay, so expectations about connectivity. This is a difficult question. Our connectivities range from around a few percent to maybe 30% for most of the elements you saw in that matrix. Our expectations are roughly in line with what we see based on what we've seen from previous literature. There are a number of experiments that in the past have shown wildly different results in those, much lower or sometimes much higher, and we think that those are generally accountable by differences in the methodology and often in the differences in the model systems as well. So I think we're reasonably close, and we're trying to do a pretty good job of characterizing the synapses that we're missing in those experiments. What was your second?
I think you're getting most of the actual connections.
I think that we're getting most of them, and I think that we're doing a good job of paying attention to where we're missing them.
The second was, are you finding any architectural principles in these
I would say that the architectural principles that we've seen so far are really just very similar to what we've seen in the past. We did see some interesting things that haven't been seen before, but for the most part, the connectivity is basically what we expect from previous literature. We're refining that picture and finally putting everything together in the same place.
Yeah, I had two questions actually. I wanted to ask you. One is, it seems it would be extremely valuable to relate the different synaptic physiological properties that you're observing to the constituents that are present at each synapse. And I know you have anatomists here, one of whom sitting next to me actually, who does this sort of stuff. And so I wondered, have you been envisioning taking this material that you've actually recorded from and then doing that?
Absolutely. We don't have concrete plans to do that at this moment, but within this institute, we have a very unique opportunity to make that happen, to take synapses that we've characterized physiologically and then move them on and look at their anatomy to establish that correspondence.
And then the second question, which I think you could only do in mice, is, as you know, there's a literature that shows lineage-related neurons preferentially connect to each other, and these are, again, experiments that in principle you could do to compare the connectivity between lineage-related and non-related.
Yeah, this is another excellent point. This one, we also don't have concrete plans for this, and this is one that we've kind of swept under the rug. That would be fantastic for us to look at, but I don't think that's in our immediate plans.
Any other questions?
Thanks. I was just wondering, it seems like from the work you've been doing, all the probabilities of having connections between different cell types is sort of averaged over a wide area of the cortex. Is that right? Or from a bunch of different subjects?
All of the work that we showed today is from V1.
Okay.
We have done a little bit of work in other areas, and we do see small differences. But for right now, we're just focused on primary visual cortex.
I guess I was just wondering, because we know that connection probabilities and connection strengths can depend on activity over a long time scale with like long-term potentiation or long-term depression. Is there some way that you can account for that in terms of trying to build a model of the connection probabilities?
This is an excellent question, and my answer to you, unfortunately, is going to be the same as my previous 4 answers, which is we would love to be able to do these experiments, and it's not in our immediate plans. There's a lot that we still need to do to complete this picture.
I'm curious, because you're doing this in human and mouse, do you have any sense at this point, maybe it's premature, about the similarities and differences for the synaptic connectivity and synaptic function between the 2 species?
That's a great question, and it is a little bit premature for us to comment on that, but I can give you a couple of interesting examples, and one is the connectivity that we've seen in human experiments has been quite a bit higher than we've seen in the mouse. Is that consistent? Another interesting thing we found is that stimulating an individual neuron in human cortex can actually cause other neurons nearby to fire spikes, whereas in mouse, they almost always just provide sub-threshold inputs. So that was something sort of unexpected that we came across.
Just a quick question. How do you account for, let's say, severed connections, and maybe you've got a bias towards connectivity at the soma or connectivity that's not as long range with those experiments, and also with the optogenetics? How do you try to prevent activation of axons that might be going over the soma?
What was the first half of the question?
Severed connectivity.
Severed connections. Mm-hmm. Okay, so yeah, in these experiments, we're looking at chunks of brain tissue that have been resected from the living brain. So definitely we are cutting connections. We try to avoid the effects of that by patching deep inside the slice. We are still going to be losing some connections. And so part of the way we're addressing this, though, is by having all of these different projects working together in parallel to characterize connectivities. So we've got a project looking at connectivity using EM in a large chunk of tissue. And so basically by comparing the results that we see to what we see in that project, we hope to be able to get a much better sense of really how many connections we think we're missing from that. Do you want to talk about axons?
Yeah, so in a couple ways. The first is that we're really trying to tailor the power of the stimulation we're using so that it reliably causes spikes in the soma and won't cause spikes in the axons, which tend to have slightly higher thresholds for optogenetic stimulation. Another way is just testing different expression methods, and so soma localizing our channels, so only the opsin is expressed in the soma where we're targeting. It's not out in the dendrites or the axons.
The relationships like the, I guess the SST cell to VIP was a facilitating synapse. Is that a, I don't know, how hard and fast a rule is that? And is the previous experience of the animal, I mean, where these neurons were in the map, or I guess if you were to go in vivo, would you expect that ever to invert, or are these strong, I guess, chemical determinants of this facilitation or depression?
That's a great question. So we have a reasonable expectation that the dynamics of the synapse, the fundamental dynamics, do not change very much as we go from in vivo to in vitro, because that's mostly determined by the sort of the molecular state of these synapses. We hope that it doesn't change in the couple of hours. However, the firing activity in vivo is very different, right? And that puts the synapse in a very different state. So that's actually the reason that we need to characterize the dynamics of these synapses under different conditions so carefully, so that when we put these synapse models into a larger network model, it can actually behave the way we expect it to in vivo. I think there was another part of your question that I missed, though.
No, I think you, I think that addressed.
Okay.
Yeah, along that same line, I thought it'd be kind of interesting to combine the physiology from the other team members working on recording the activity of the neurons in a behaving mouse, and then to play those action potential patterns into the presynaptic cell to see if the dynamics, which you're characterizing simply as facilitation or depression, actually predict what the synapse does in a kind of real-life pattern of activity.
That's a very good point. And we have talked about doing these experiments as well, where we even take tissue that has gone through these imaging systems and bring them down into the physiology experiments. The difficulty with that is that we don't know the connectivity from those experiments. So we can replay these firing patterns, but we don't necessarily know which of those are connected to which. So that makes it difficult for us to determine how to replay all of that activity into the network during the experiment. But that's a great idea, and we have spent a lot of time talking about what are the correct firing patterns to play into these neurons in order to adequately sample the dynamic range of the synapse.
Okay, maybe just one more question.
Okay, so the question is, when anecdotally we see this effect that in human tissue, when we stimulate a neuron, it's more likely that that's going to cause other neurons to be activated. And the question is that borne out in the EM data? And unfortunately, I have absolutely no idea. But
targets more inhibitory neurons than chance, while in cats and monkeys, it targets much more excitatory. It targets cortical to chance. We have much more connections to excitatory.
So I'll try to summarize what Nuno has just said. You're going to hear from Nuno later today. He's going to talk about the EM project. But the first was that excitatory synapses in mouse are smaller, so we expect something like this. And the other was that there are differences, known differences in the patterns of connectivity with inhibitory interneurons. Is that? Okay. Oh, sure.
Right. So let's thank the Synaptic Physiology team again for a great talk and great question and answer session. Thank you.